everyone. My name is Janaki from Strand Scientific Intelligence. Welcome to our webinar on Genome Browser in Avadis NGS. In this webinar, I'll walk you through the features of the Genome Browser as part of Avadis NGS. Avadis NGS is our offering for analyzing DNA-seq, RNA-seq, and ChIP-seq experiments. Strand itself is a 10-year-old scientific software company with a history of building products for analysis of omics data. In particular, GeneSpring, one of the leading products, is for analysis of microarray data. With this brief introduction, I would like to get started with the webinar on Genome Browser. At any point in time, if you have questions, please use the chat window to type in your question, and I'll try to provide answers towards the end of the webinar. For those of you who are already using Avadis NGS, you probably should know this, but for those who are new to the software, Avadis comes with a fully embedded genome browser that is extremely feature-rich and easy to use. The fully embedded part should be pretty obvious, but I hope that by the end of this presentation, you will take my word on the feature-rich part and the easy-to-use part as well. What this means is that you will not need to export data or your results and take it to a different genome browser outside Avadis, like let's say the UCSC browser or the IGV browser. On the contrary, it's actually quite easy to bring in data from external sources by just dragging and dropping it into the Avadis genome browser. The underlying philosophy of the genome browser in Avadis is to enable simultaneous visualization of your raw sequencing data, of algorithm or analysis results, and public annotations. All of this simultaneously in a single view. Most other browsers provide you the ability to visualize either one or a subset of these three, and others that do provide all of them, not sure that they are that easy to use. Let me show you what I mean by simultaneous visualization of data, results, and annotations. This is a screenshot of the Avadis application window. To the left, you see the project navigator that contains multiple experiments. 3 of those experiments are currently open, and one of the experiments, the NRSF REST experiment, is the active experiment. On the right, you see the workflow browser, and you see the workflow browser for this experiment. And since this is a ChIP-seq experiment, the workflow steps specific to ChIP-seq analysis are shown in the browser. The centerpiece is where the genome browser is shown. Other visualizations also share this central area along with the genome browser, but for now we only have opened the genome browser for visualization. The topmost track shows the raw sequencing data, in this case the forward and reverse coverages for the ChIP-seq sample. It also shows the reads in that location. The next track shows the result of an analysis step, in this case the peak finding algorithm. The small green colored region that you see at the center of this track is the detected peak region with a score of some 350 odd. The last track shows public annotations. In this case, it shows UCSC transcripts, and you can see a gene close to this peak that has been found by the algorithm. When you load samples into Avadis NGS to create an experiment, the first visualization that is shown at the end of the experiment creation process is that of the genome browser. The genome browser at this stage shows only the raw sequencing data and the annotations. Note that at this stage, no algorithms have been run, and the algorithm results can be added at a later stage. This particular picture shows the state of Avadis when this RNA-seq experiment was first created. This is an RNA-seq experiment performed on 2 cell lines, CD4 and Jurkat. And since the experiment was created with the EDGE18 build and with UCSC transcript annotations, the default annotation tracks shown in the Genome Browser is the UCSC transcripts. The data track shows the coverages of the 2 samples, and this is the default view when you created the experiment. But when you open this, if you close this experiment and close Avadis, and at a later date open the same experiment, this is the same default view that would show up. At this stage, when you've just created your RNA-seq experiment, it's recommended that you spend some time looking at the coverages from each of the chromosomes. That will serve the purpose of spotting any obvious abnormalities, like let's say a very high coverage region in an unexpected place, or let's say regions of very low coverages. In fact, for an RNA-seq experiment, if you are aware of, let's say, genes that are known to be differentially expressed between, in this experiment, let's say we know a particular gene that's supposed to be differentially expressed, you can just search for the gene and take a quick look at the coverages across the 2 samples. Here, for example, the SIX6 gene is indeed shown to be differentially expressed based on the coverage information. Yet another gene here is YBX2. And again, you can see that the coverages for this gene in one of the samples is far more than the other sample. In fact, in the other sample, it's not expressed at all. You can also see here that the coverages are neatly aligned to the exonic regions of this transcript. It so happens that there are some reads and there is some coverage in intronic regions. And in an RNA-seq experiment, if you perform novel detection, it's likely that this region would be detected as a novel exon for this gene. When you create a DNA-seq experiment, this picture similarly shows the state of Avadis at the end of the experiment creation. This experiment that I'm showing you is a whole exome experiment performed on a normal sample and a tumor sample, on a normal tissue and a tumor tissue of a tongue cancer patient. Since the experiment was again created with EDGE18 and UCSC transcript annotations, the default annotation tracks shown in the Genome Browser include the dbSNP 130 and the UCSC transcripts. Note that unlike in the previous RNA-seq experiment, here the dbSNP annotations are also shown by default, since it's quite likely that in a DNA-seq or a whole exome experiment, one is likely to be interested in SNPs. The data tracks again show the coverages of the 2 samples, and as usual, since no algorithm has yet been executed, there are no analysis results shown. Again, just like the RNA-seq experiment, at this stage, it's recommended that you spend some time looking at the coverages from the different chromosomes and try to spot any obvious, let's say, abnormalities. For example, if I zoom into this particular gene, the CASP7 gene, one can easily check that the coverages from the 2 samples are extremely well aligned to the exonic regions of this transcript. Just like the previous RNA-seq example that I showed, the key idea here is that by taking a look at the genome browser and the coverages across chromosomes and across the samples, we should aim to get a feel for what the data is saying. This example here shows the genome browser in a ChIP-seq experiment. This experiment was performed to analyze the transcription factor regulatory effect of the NRSF rest protein. Here you can see tracks from the ChIP sample and the control sample, and as usual the UCSC transcript annotations, because that is what the experiment was created with. If you take a look at these coverages, it looks satisfactory. There seem to be peaks, some tall peaks in several parts of the ChIP sample, and the control sample largely shows low coverages. Indeed, if you zoom into some of the tall peaks in the coverage of the ChIP sample, you'll see nice peak binding sites as shown in this example. The control sample here does not have any peak, and this peak is close to the gene shown here, and it's possible that it regulates the transcription of that gene. Note that unlike the DNA-seq and RNA-seq experiment, the default view in the ChIP-seq experiment shows forward coverages, reverse coverages, and the total coverage. All of these are shown in distinct colors, and the total coverage is shown with a thin black line. The idea here is that in a ChIP-seq experiment, one is largely interested in looking at the peak regions where the forward and the reverse coverage are expected to be slightly separated by the size of the binding region itself. Now let's take a look at this other ChIP-seq experiment. And if you take a careful look at the coverages, it looks a little fishy. There are peak regions that you can see in the ChIP sample. In fact, this experiment doesn't have a control sample, and you can see peaks in the ChIP sample. But there also seems to be significant coverage uniformly across the rest of the genome. And if you zoom into some of these regions, you will notice that there are these huge coverage regions. This region, for example, shows a coverage of 80, but all these reads are largely duplicate reads. This would immediately suggest that it may be a good idea to run a filter to remove duplicate reads from this sample before continuing with the analysis. Okay, so that was a quick overview of the genome browser in Avadis NGS and the initial view that you get when you create an RNA-seq, ChIP-seq or DNA-seq experiment. Before we get into details of the other features of the genome browser, this would be a good point to understand what I'm calling the anatomy of the genome browser. As you would have noticed, the genome browser has a number of these what are called dockable sidebars. They display data in various different forms. The chromosome view shown here is one such dockable sidebar, and in the default state, it shows the cytoband of the chromosome that is currently being visualized. If you take a careful look at this chromosome view, there are these two small buttons at the top right corner of the chromosome view. There is this title bar, and there is this tab which says chromosome view. It is possible to rearrange this sidebar and the various other sidebars by just dragging by using these buttons and by dragging and dropping the title bar or the tab view. So let's do that, and this is a view that you can get from the default view. In this view, all the sidebars have been docked in places to give you a feel for the anatomy of the browser. The chromosome view that we saw in the previous slide has been dragged and displayed on the right side of the Genome Browser. As you can see here, the cytoband has also automatically changed its orientation, and this is much like viewing a photo, let's say on an iPhone, and when you turn it around, the orientation of the photo changes automatically from landscape to portrait. This sidebar on the left is the genome view. It's called the genome view, and it shows us a picture of all the chromosomes of the organism, or more specifically the build that is being used. The chromosome that is being currently displayed is highlighted with a green selection border. The region view is the centerpiece, and by far this view is where most of the action happens on the Genome Browser. Data is displayed in the region view in the form of tracks in the main visualization section. A track is essentially a plot of the chromosomal location on one axis and some chosen data on the other axis. The data could be reads, the data could be results, or it could be annotations. The region view is split into two panels by this thin divider here. By default, the reads are shown in the top panel, and algorithm results and annotations are shown in the bottom panel. In each of these panels, any number of tracks can be shown, and all of them can be stacked one on top of the other with a scroll bar. These tracks can also be resized by these separators, by dragging these separators up or down, and it's also possible to move a track from the top panel to the bottom panel or vice versa, using this small triangle button here. The next view that one would be interested in is the spreadsheet view, and the spreadsheet view shows data from the active track of the region view. If you notice the region view, there are 4 different tracks, and one of these tracks is highlighted in green and is the active track. The data from this track, it could be reads as is shown here, it could be algorithm results in the case of this track, or annotations in the case of the last track. That data is shown in a tabular fashion in the spreadsheet view. At the bottom right corner, you can see the annotations corresponding to the appropriate build of the organism of interest. These annotations can be again visualized in the region view by just double-clicking a particular annotation or single-clicking an annotation and clicking on the Add button at the bottom. At the bottom left corner, you can see what is called the search results panel. I had mentioned earlier in the RNA-seq experiment that you could search for a gene, and that search is performed from this text area in the toolbar. When you search for a gene, or you could search for other annotations as well, like let's say dbSNP entries. The results of the search are shown in the search results panel. So these 6 different views together form the anatomy of the genome browser. Now, just by themselves, if they are static, that would not be of interest. So obviously these different views interact very closely with each other. Let me show you how these interactions are performed. It's possible, for example, in the genome view to click on any of these chromosomes, and the result of that would be that the chromosome view would change to show that particular chromosome. The region view would change to show data from that particular chromosome for all the tracks. Given that this track is the active track, the spreadsheet would in turn show data from the active track of the changed chromosome. Now let's say we've changed to a different chromosome. Within that chromosome, you can again click and drag on in the chromosome view, and the result of that would be to zoom the region view into that region, and the spreadsheet would again show the data from the active track in that particular region. You can also zoom in from the region view, and you can also interact with the region view from the spreadsheet. For example, you could click on one of the rows of the spreadsheet, and that particular item in this track would be highlighted across all the tracks in the region view. You could double click on a row in the spreadsheet view, and the region view would automatically zoom into the location of that particular item. Okay, there are other interactions that one can perform. For example, with the region view, let's say you want to change chromosomes. There are multiple ways to do this. You can use the genome view to switch from one chromosome to the other by just clicking on that chromosome. It's also possible to change chromosomes by just typing in the name of the chromosome in this text area. And it's also possible to change the chromosome by just using this drop down here. Using the drop down is usually the most convenient and most often used method. The other region view interaction that is most commonly performed is zooming into a region of the tracks. There are multiple ways to do this again. You can use the toolbar buttons at the top. There is one to zoom in, one to zoom out, and a third one to reset zoom to the whole chromosome. You can also click and drag on the chromosome view as mentioned earlier. And when you do that, the region view shows data from just that region of the chromosome. But the more often used way to zoom in is to double click on a track. For example, let's say you are interested in this particular region of the chromosome. You just double click on the track at that region, and the tracks would zoom in centered at that location. The other very often used way to zoom in is to shift, click, and drag on a track. When you do that, you'll see a light pink band showing the region that will be zoomed into. When you use double click to zoom into a track, you can use shift double click to zoom out as well. The genome browser in Avadis does what is called shows data in what is called semantic zooming. So as you zoom into to a region, this track here, which was showing only coverages, automatically starts showing coverages as well as reads. At this stage, the reads show only high-level read information. For example, the CIGAR of the read, the alignment or the alignment score or the mapping quality of the read. If you zoom in further, you can start visualizing the individual bases on the reads. By default here, for example, a read is shown along with bases that are that have a mismatch with the reference at that location. For example, this read here has two mismatches towards the end of the read. And you can again mouse over the read, in fact, mouse over each of these locations to get information about the read in all, about the overall read, as well as about the base at that location. Okay, the next common interaction that one does with the region view is to pan through the tracks, pan through the genomic locations. There are three ways to pan. You can just click and drag a track to the right or to the left, and it would just pan seamlessly to that in that direction. There are other attractive ways to do the pan. For example, on a track that doesn't have scroll bars, if you are using a Windows or a Linux machine and your mouse has the mouse wheel, you can just scroll the mouse wheel to pan the track. On a Mac machine, you can use a two-finger gesture on a track without scroll bars to pan the track to the left or the right. Okay, so those were some of the high-level interactions that that one should be familiar. The various components of the Genome Browser and the interactions between the components, and some common interactions with especially the region view component of the Genome Browser. Let's move forward and take a look at how you would look at results from within the different experiments in the Genome Browser. For example, in a DNA-seq experiment, you may be interested, if it's an exome experiment, in looking at the target regions and the coverages in those regions, or looking at SNP results or structural variant results. In an RNA-seq experiment, you may be interested in looking at interpretations, meaning, let's say you have 10 samples or let's say 12 samples with 4 different conditions and 3 samples in each condition. You may want to compare the first condition and the second condition and look at only those samples in the Genome Browser. It's possible to do that seamlessly. You may be interested in looking at novel genes or novel exons in your RNA-seq samples. You may be interested in looking at novel splice junctions or gene fusions. In the next few slides, I'll show you how to visualize some of these results seamlessly in the Genome Browser. In a ChIP-seq experiment, like I mentioned earlier, you may be interested in looking at peak regions or motif regions. Apart from these results that are specific to each of these experiment types, there are other common kinds of objects which come up in all these experiment types, and those include read lists, filtered read lists, region lists, and gene lists. It's possible to visualize all of this very nicely in the Genome Browser. Let's take a look at a few of these carefully. By the end of this, you should be able to get a very good hang of how to use the Avadis Genome Browser and the features that it provides. So this experiment here is an exome experiment. The bottom left corner shows the experiment navigator for this experiment, and it also shows the region list for the target regions. This was the target regions here are from the human exon kit from SureSelect, the G3362 kit. What you can do is just drag and drop this region list into the Genome Browser. And then you can zoom into any particular region of the Genome Browser to look at some of the target regions. In this particular case, I've zoomed into the Cas7 gene like before. And if you recall, the coverages from these two samples are very nicely aligned to the target regions itself. Okay. What you can then do is, let's say, so you can then use the navigator buttons here, and these buttons are extremely handy when you're looking at the Genome Browser. You use the next arrow or the previous arrow here to go to the next region in this track. So as you can see here, there are some 7 odd target regions being shown overlapping the Cas7 gene. When you click on this next arrow here, the view automatically changes to the next region of to the next target regions, and the regions that were overlapping the Cas7 gene have shifted to the left. You can now again see that these target regions and the coverages are nicely overlapping, and you can keep exploring the target regions in your DNA-seq experiment in this manner. Okay, this is yet another example of viewing results in the Genome Browser. In this particular case, we want to look at the results of SNP detection in this DNA-seq experiment, and we want to visualize that in the Genome Browser. If you look at the experiment navigator here, this node here is the result of the SNP detection algorithm. And you can again drag and drop that into the Genome Browser, and that would show up these two tracks. The defaults in these tracks are also set up nicely. So the labels that are shown at some of the places are the SNP calls themselves. The colors that are shown for the SNP calls indicate are based on the decibel score for the SNP call. Green indicates a low decibel score, and red indicates a high decibel score. Now there are several ways in which you may want to go forward from here. Note that at this stage we are looking at chromosome 10 and the entire chromosome. One thing that we can do is to just do the regular zoom operations, and let's say you can zoom into this particular region that shows an interesting red SNP, which means a SNP with a high decibel score. That would look like this. When you zoom in, you can see in this particular region there are 4 different SNPs. 2 of them are homozygous and 2 of them are heterozygous. There are 3 SNPs and there is a deletion. 3 of them are novel SNPs, and 1 of it is known from dbSNP. This SNP here is known from dbSNP, and these 3 SNPs here are novel SNPs. And you can see a lot of other information about the SNPs themselves. This particular SNP, for example, is a deletion, and you can see the nice drop in coverage at that location. This SNP is a homozygous SNP, and you can also see this uni-colored bar here that shows that most of the reads here have a mismatch with the reference, and the color of the bar indicates that the mismatch base is actually C. These 2 SNPs here are heterozygous, as you can see from the multicolored bars here. It's also possible to interact with the SNP results in multiple other ways. For example, you can sort by one of these spreadsheet columns and then just double click on a particular row to inspect that particular SNP. There is yet another interesting way in which you can inspect the results of this SNP detection algorithm. To do that, you right click on this track and go to the filter options, launch the filter options dialog. Here you can set any filter condition on any of the properties of the SNP results. Here, for example, I've set a filter condition to say that I'm interested in novel SNPs of the homozygous kind, and I'm not interested in deletions or insertions. I'm interested in SNPs. When you perform this filter, you'll see that this track that was a large clutter of SNPs across the entire genome is filtered to the narrow list of SNPs that I am interested in. You can again zoom into individual SNPs and inspect them carefully. You can double click the spreadsheet view and inspect a particular SNP more carefully, and so on. Okay. Excuse me. Let's take a look now at how to visualize large structural variations in the Genome Browser. This is a different experiment where structural variation has been run, and by now you should be familiar that you can just drag and drop this object into the Genome Browser, and you will see the regions that were detected as a result of the SV algorithm. The colors and the labels are self-explanatory. There are insertions, deletions, and other kinds of SVs. The color here in this case is based on the number of deviant reads that supports that particular structural variant. As usual, you can interact with this in several ways. You could just zoom into a particular region. You could sort the spreadsheet, double click on one of the rows of the spreadsheet, and inspect the structural variant. In this particular case, I have zoomed into one homozygous deletion that was found in one part of chromosome 1. To be able to look at the reads in this form, you need to right click on the reads track and open up the properties dialog. In the properties dialog, you need to enable connection between the paired reads. Once you do that, the Genome Browser shows paired reads connected by a dotted line. If you notice, there is also a color difference between the reads that overlap this deletion and the reads in other locations. Most of the reads here are light gray, and most of the reads in this region are a dark grayish color. This dark gray color signifies that these paired reads have been separated further than the expected insert length, and that signifies a deletion. It's also possible to verify that again using a filter. So you can say that on this read track, you want to look at only reads that have a status of mate far. That is, you want to look at this track with only the mate far reads shown. And now it will become immediately obvious that in the midst of several of the other reads that were being shown, there is a huge cluster of mate far reads here, which signifies a deletion. The pairs on this end come close together, and the deletion itself is in this particular area. Okay. So let's move on to take a look at how one would visualize results in an RNA-seq experiment. Here I'm looking at an experiment that has an RNA-seq experiment that has been created with 5 different samples. by default, the coverages from all these 5 samples are shown in the region view in the Genome Browser. And what I want to do now is to inspect the novel genes or the novel exons that were detected by the quantification algorithm. To do that, I take the output regions of novel detection, I take the output genes of novel detection and just drag and drop them into the Genome Browser. Note that this is a region list, which means that it just has every individual item in this region list is just a region of the chromosome of the genome. This is a gene list or an entity list, and every item in this is actually a gene, a known gene. Rather, in this case, every item in this list is a novel gene. When you drag and drop these 2 items into the Genome Browser, you can immediately use all the techniques that we've been using in the past to zoom into one of the genes, one of the novel genes that was detected. And here you can see that this gene comprised of these novel regions was detected as was called out as a novel gene by the novel detection algorithm. And again, you can see that so Avadis today doesn't compute a new transcript at this location, but it just calls out a novel gene, and that is shown by this location. And in the future, when we have the ability to update transcript models, one can imagine that these regions that form part of the gene would most likely be called as the exonic regions of that transcript. Similarly, it's possible to look at novel splice junctions in an RNA-seq experiment. And the way to do that is again similar. The novel splice junction created this particular output in the experiment navigator. You just drag and drop it into the Genome Browser. And in this case, in chromosome 1, there are just 2 novel splice junctions detected. This is the active track in the region view, and the spreadsheet view shows information about these 2 novel junctions. Turns out that there are roughly 35 reads that substantiate this particular novel splice junction call, and when you zoom into that splice junction, you can see that this gene ZNF691 has 3 different transcripts. But if you look at the coverages across the samples, you'll notice a lot of solid lines between this peak here and this peak here, and that is true for multiple samples. What it means is that there are reads that map to this exonic region, and there are parts of that read that map to this exonic region. And this splice junction is currently unknown to UCSC transcripts. A small thing to note here is that earlier when we visualized SV results, the connection between paired end reads was shown with dotted lines, and the connection between spliced parts of a read are shown in solid lines. Okay, there are a few other small things that that you may be interested in looking at here. So there are a few other buttons here. I talked about the triangle button in the beginning that is used to move this track across the panes of the region view. The navigator next and previous buttons that are used to go to the next region in this track. For example, here if you just click on the next button, you would go to the other, you would automatically reach this splice junction, and you can visualize it again in a zoomed in mode. There are also these maximize, minimize buttons, and I've used the minimize buttons to minimize some of the samples. Recall that I had 5 samples in this experiment, and I've minimized a couple of samples. Okay, moving on. Here we are trying to visualize in this RNA-seq experiment gene fusions. As usual, the gene fusion results show up here in the navigator as a result of running the algorithm. The gene fusions are shown, there is one region list for the gene fusions from each of the samples. You can, as usual, drag and drop that region list into the Genome Browser to visualize. When you do that in this particular case, you see that there is only one gene fusion detected in chromosome 1. When you carefully inspect the results in this sample, the gene fusion results in this sample, you can do that by just double-clicking the result node here. You see that the total number of regions, gene fusions that were detected were 26. So one way to check where these gene fusions are lying is to use what is called the genome view to display that information. So you right-click on that track, on the gene fusion track, open up the properties dialog, and you notice that the gene fusion track is being shown in the region view, but you also want to see, let's say we also want to see that in the genome view. So you just check this particular checkbox here, and then what happens is that the regions of this genome view, the regions of this track are shown in the genome view across all the chromosomes. This gives you a quick snapshot view of where all your gene fusions lie. Once you have this, now you can say that I'm interested in looking at gene fusions that fall in, let's say, chromosome 11. Click on that chromosome 11 to make the region view focus on chromosome 11. There are a few gene fusions here, and clicking on this track shows the tabular information for the gene fusions, and there are, as you can see, there is a gene fusion between these two genes with a large number of paired reads going across them. If you click on those two rows, these two genes are highlighted, these two regions are highlighted, and you also see this thin gray line that shows those regions. You could zoom into one of those regions, and then you can see again that that there are several reads here that have dotted lines going across this gene, right? These dotted lines stretch across the gene to further away. If you click on one of those reads, you can see that this read itself had a start and end close to 800,000, but the mate location is far away at roughly 100 million location. And that is how you can verify this gene fusion happening here. Okay, so that largely covers how you would visualize results from the individual algorithms in the Genome Browser in conjunction with your raw sequencing data and the annotations. So at the end of the day, the Genome Browser would enable you to make sense of the algorithm result in conjunction with the other data that you have. A few other small features before we wind up that I thought would be interesting to showcase. There is this small toolbar button here at the top left. You click on that to get the Genome Browser into a full screen mode. What that will do is very similar to this PowerPoint slideshow mode that we are seeing here. The Genome Browser would get into a similar mode that gives you a lot of real estate to carefully visualize and inspect your tracks of interest. If you're interested, you could also change the orientation of the tracks, change it from the usual horizontal orientation to this vertical orientation. This is sometimes useful if you have a large number of samples Usually screen sizes are long on the horizontal side, and in that case you would like to lay your tracks in a vertical fashion to accommodate a large number of samples. There are several other features that I haven't been able to cover, but once you start playing around with the Genome Browser, you'll find a lot of these interesting features. That come in very handy when you are looking at your data. I think with that I would like to conclude this webinar. You can avail of further information from our website www.avadisngs.com. If you haven't downloaded Avadis and tried it yet, you can download the installers for your appropriate platform from the website. You can also use this link here, start an evaluation, to get a license key for 20 days that is fully featured. And if you have any questions, feel free to contact us at avadisngs.sales@strandsi.com,